We're going to be discussing the basics of retrieving your sequence data that you sent off to identify your bacterial species. In this section, we'll be discussing the chromatogram. Now, the company returns two files with the extensions .ab1 and .seq. The .ab1 file is our chromatograph and can be opened in Macs or PCs. PCs will have to use a Chromas Lite version of Chromatogram software, while the Macs can use 4 peaks. Once you've downloaded this program, you can open your chromatogram, and what you'll see is that the chromatogram is a graph with 4 different colors. Each of these colors represents a nucleotide. So green for A, blue for C, black for G, and red for T. As we scan across the chromatogram, this is our entire sequence, we see that there is a single peak per nucleotide in this region. And this is what we consider a clean, clear chromatogram. There's a single peak with very low background. And this is important because this gives us confidence that the sequence we are given is the sequence found in this sample you sent out for sequencing. Now, when we talk about an unclear sequence, this is typically found if in a sequence at the beginning and end. So let's scroll to the end, and you can see when I talk about what I mean by an unclear sequence. Here we have peaks that span more than one nucleotide without continuous peaks. We also have multiple peaks in the same region. Let's see if I can find one that signify more than one nucleotide being found at the same time. This is the unclean region, and it is important to note these regions on your chromatogram and on your sequence so that when you take these sequences to blast search, you will remove this area from the sequence before blasting. The chromatogram is a nice visual example of your sequence, but to do a blast search, we have to use the written sequence and the .seq file, which we'll talk about in the next